Okay, can you guys hear me? Yes, you can. Okay. We're doing a little bit better in terms of presence. That's good. Because I realized that profs don't record because people don't come. So it's okay. We'll continue recording if you guys keep coming, okay? What did I want to ask you? First, just a heads up for the people that are registered with ACSD. ACSD, yeah, exactly. So for the midterm and the final, you'll be doing your exam by yourself at the office of ACSD, okay? That means that you'll get the time required for the midterm and the final plus the 33% or 50% that you need, okay? That's the first thing. For quizzes in class, it's a bit more complicated because I basically have two options. Either I make you do the quiz by yourself at the office and then make you run back to the tutorial to not miss anything, or I give everyone the same time, and that's the time that you would get with the additional extra time you require. So the quiz should take about 20 minutes. The person that needs the most extra time requires 50% time more, and so I'm giving you 30 minutes to everyone, basically, okay? This is how they do it at the ACSD. I don't find it 100% good in terms of fairness to the other people, but we have to do it this way, right? Because if you need 50% more time, that's okay, but then if I'm giving everyone else more time also, then, you know, it's a gray zone, but they told me to do it this way, so we're going to do it this way, okay? Make sense? For the midterm and final, I will be running between the ACSD office and the classroom. I'll have the TAs with me to invigilate, so they'll be able to help you, okay? How was the quiz? Not bad? Easy? Okay, so the TA told me, one of the TAs told me that she went through the copies very fast, and she realized that actually most people did well, just one or two that had like one or two mistakes or something like this, okay? So it's probably gonna stay like this for the whole semester. We'll see depending on the topic, okay? Good, what else? I talked to the publisher, so they allowed me to post all the solutions to the problems in the book. So what I'm gonna do is post them Wednesday after 5:30 p.m. The reason for that is to allow you guys to work during the tutorial so you can practice, okay? Instead of just having all the answers and then just waiting for the end of the tutorial, okay? Make sense? Good. Any concerns, problems with the TAs, with the tutorials, teaching style? No? Everything's good? Okay. Cool. So today we're going to do two things. First, I'll start with lecture 5, and then I'll go back to lecture 4 just to review a few concepts that we didn't do last time. And then that's pretty much it. So let's get started. Okay, so we start by the map. So this is something crazy that I actually saw maybe yesterday or 2 days ago, I think. And I was actually shocked, okay? Because I was looking at recombinant maps and all that, and, you know, TikTok's algorithm understood that and started showing me stuff about maps, okay? So apparently, this is actually super, super wrong, okay? And I'll tell you why. First of all, I don't know who drew this, but Africa is supposed to be almost as big as the whole America, okay? So this is just something I'm sharing that's unrelated to the course, okay? But it's a good introduction because we're going to talk about maps today, okay? So I was thinking actually, like, why is this like wrong like this? And apparently all maps that were drawn are wrong, okay? Africa is much bigger than that. In fact, Algeria, where I come from, is supposed to fit 4 times in Canada, okay? But it can fit like 20 times. And Brazil, if you add it to France, it should be Canada complete, okay? So this is pure bullshit, okay? This is not good stuff, okay? One extra fact, okay, about Algeria, because pride, okay? One of the oldest civilizations is found in Algeria. There's a city called Sifar, so it's one of the oldest civilizations that dates back to 12,000 years ago. They're called Amazighs, and I'm one of them. So that's basically the natives of Algeria. Okay, cool. So enough about maps. Let's talk about real maps. Genetics, okay? So in genetics, we use maps to kind of link genes together, see where they fit on the chromosomes, okay? It started very simply as one map that links 2 to 3 genes, and then it turned into the whole genome, okay? And I'll just give you an example online so you have an idea about what it actually looks like in terms of scientific research. This is not for your exam, it's just an example for you guys to see that what you're learning is not useless, okay? So if you go to UCSC browser, and then you click here, and then you go to genome. It's not as cool as the things we're learning. You're probably not going to get anything, but I'll explain some stuff to you. So if you click here, for example, and you write CHRY, so the Y chromosome. Just 1 second. So you can see all the genes that are found on the Y chromosome, okay? Those in green means we don't know what they do. Those in blue, they have a name, and we know what they do, okay? For example, DAS3. So DAS3, it says that it's important for spermatogenesis. So if you don't have it, you don't make gametes, okay? Things like this. And then you can see that the genes are basically, this is one thing that's bad. If you want to see on the X chromosome, you can do the same, and then you will see all the genes on the X chromosome, okay? And then from here, you can, have fun and click on each one of them and see what they do, okay? This one is going to take time to load because there's a lot more genes, okay? Cool. So that's pretty much it for the research side. Actually, I use this tool a lot because I try to figure out where a protein of interest binds to the genes, and then I'm trying to mutate that to prevent cancer, okay? Looks very boring, but trust me, it's interesting, okay? Do you guys know this? You've seen this? Do you guys see the colors? You see the number? Anybody not see the number? Really? Are you red, green, blind? You are? Okay, that's interesting. Do I have to change my slides to make them fit, or you're good? Okay. So I'll still try to do it. You know what? Why not? So basically it's blue and yellow, right? That I have to adjust, I think. Okay, cool. Okay, that's perfect. So this is things out of your mind, Okay, so we'll see at the end of the lecture, why do you pass this test at the optometrist, and how is it interesting in terms of genetics, okay? And why does red and green blindness go together? And why does it happen more in male than in female, okay? Actually, if we go back to the genome browser and we write OPN1LW, so this is a gene that receives light of the long wave length, and then it's found on the X chromosome. So if you get one bad copy as a male, then that copy is that copy, okay? So that's that. Now the boring stuff, history. Okay, so when Mendel was working with his peas, he actually studied more than just those 7 genes. He actually made combinations where he found that the inheritance 9 to 3 to 3 to 1 does not actually work. And what he realized, and then Punnett and Bateson, these guys took this research again, and they tried to figure out why, and they realized that 2 alleles always follow each other, okay? And it's almost like if they were physically linked, right? And so this is just a prehistory of how they found that 2 genes sometimes are linked on the same chromosome and sometimes are on separate chromosomes, okay? And then there's this guy called Morgan, who did the same thing, studied 2 genes for the Drosophila, the fruit fly, and he found the linkage hypothesis, okay? So these genes that he studied were the eye color and the length of the wings. So PR is for purple. So if you have this PR here, the recessive allele, that you'll end up with purple eyes. And if you have the vestigial recessive allele, then you'll have short wings, okay? And then the positive one, the wild type one, is the long wings with the red eyes, okay? So this gives red eyes. So he did the same thing. So just now you have to switch your thinking from 2 different chromosomes to the same one, okay? And, okay. So let's go through his experiment that he did, and then we'll go through the theory, and then we'll do some problems, okay? So for his experiment, he took 2 flies that were homozygous for so one fly was homozygous recessive for the purple eyes and small wings, and the other one was wild type, so long wings, red eyes. He crossed them. So before crossing them, the gametes are these, and then fertilization happens. You get the, let me just get the laser. Yeah, so, and then you get a dihybrid, okay? So this is typical of all the genes that we follow, right? So you get your parental, gametes, dihybrid, and then for the dihybrid, he went and did a cross, okay, a test cross with a double recessive. And he obtained these numbers here. Technically, if the genes are on different chromosomes, you would expect independent assortment, and you would expect the same numbers here, 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 and here. So that would be like about 700 something. But he found these numbers. It seems like these two alleles go together more often than the others, okay? And so it's telling you that probably they are linked compared to the others, okay? So then he went on and measured the recombinance frequency or percentage of these two genes by adding the recombinants. So these two here are called the parental because they look like the parent, right? So the parent PR plus, VG plus, right? And then the recombinants is where you have the PR plus and then just VG, okay? Because these don't look like the parent, right? So he looked at the recombinant frequency, so you add these two numbers, divide them by the total, and gives you 10.7%, okay? So this is not your typical 50% that you would get in a regular cross for two genes that are on different chromosomes. Does that make sense up to now? Okay. So from there, he was able to think that the parental combination of alleles are held together more often than the recombinant, and so they must be somehow linked together. Okay, the inheritance of one is dependent on the other. Okay, and on the map for the chromosomes, it should look something like this. So you have your parental right here. Okay, good up to now. Okay, then he did the same experiment but changed just the genotypes of the parents. So again, he took one homozygous dominant for the eye color gene and one homozygous recessive, and then crossed it with the opposite. And he made the gametes, so those gametes just come from this here can only produce one type of gamete, so PR plus VG. Then you get your dihybrid again, and the dihybrid from there you test cross it, so you get the gametes from this one, which are these right here, and he found these proportions. Again, showing you that this is following probably a linked inheritance for these two genes. Okay, just that, yeah. The wild type is the PR plus and the VG plus. Yeah. In your book, if you read the book, this line here is written twice. Okay, that's just a mistake. Right, so there should be only one line. So I removed it from the slides, but in your book you have like 5 lines. Just forget about one. Okay. Yeah. So when you're doing a dihybrid test cross like this for linked genes, you always expect this kind of behavior. Okay. So you will always have a group of parental gametes that are parental-looking gametes that are in high frequency, and then the recombinants in lower frequency. If it was all in independent assortment, as we've seen before, you would have the same amount here, okay? But in this case, so it would be, the recombinants would be 50%, and then the parental would be 50%. But because we are talking about linked inheritance, we have to think of it differently. So you would have two equally frequent non-recombinant, sorry, okay. Yeah, two frequently non-recombinant classes totaling more than 50%, so that's the parents, and then less than 50% is the recombinants. Okay. Another way to show this is to go through the previous examples that we looked at. So if we're taking a gene, let's say A and B, so here they're on different chromosomes, and then we end up producing the gametes and the F1 here. Now this F1 can produce only 4 types. So when it's crossed with the tester, obviously can only produce 4 types of progeny, right? So it would get this from dad or whatever, and then this one from mom. and then you're producing 4 different types. The ones that look like the parents, so this one looks like this one like this one, we call them parental, and they come exactly at 25% each. The recombinants, so these are variations from the parental, they come at 25% each also, okay? When the genes are linked, now it's a bit different, because what happens is that there's crossovers that happen, and crossovers don't happen all the time. They actually don't happen, almost never happen, but they do, okay? So the parental has more chance of being passed to the offspring than a recombinant chromosome, okay? So what you would find is the parental always being more than 25% each type, and then the recombinant to be lower than 25% each. Yeah? Yeah, exactly. So, yeah, go ahead. Okay. Okay, so we're both talking about A, B genes, okay? The only first difference that you see is how they are relative to each other. So in the first diagram, we're talking about 2 genes that are on different chromosomes. Here, they're on the same chromosome. That's the first difference. Second difference is, so in this case, when you have the progeny formed, you're thinking chromatids, right? So this chromosome here comes with this one and this one and this one, okay? When you were talking about the genes that are linked, we're talking about this type of chromosomes, which are resulting from the crossovers, okay? And the third difference is the numbers here. So in 2 genes relative to each other that are on different chromosomes, the parental types in the progeny, so the ones that look like the parent, are always more than 25% each. So equal to 25%, okay? And then the recombinant are also equal to 25%. Yeah. Just one second. And then the other one, when you were talking about genes that are linked on the same chromosome, the parental will always be more than 25% each. Why? Because genes that are linked have more chance of going together than being into a crossover. Okay? Yes, Isabel. Chromosomes, sorry. Yeah. Chromosomes. Yeah. Was that your question, or did I just... Okay. How come there's 4 versus 2? It's actually 2. It's just here it's linked. That's it. Okay. Yeah. So okay. So this theory here, this experiment here does not show you why. This experiment here does not show you why. Why do these recombinants happen when the genes are linked, right? So Morgan actually proposed the theory of crossovers. Actually, his student, which was an undergraduate student. I have no idea how this guy was this smart, because me as an undergrad, I would have never been able to figure this out, okay? But this guy just went home, apparently, from the story I read in the book, and figured out the story of crossovers in a night, and then didn't do his homework, apparently, for another class. But you know what happens? Whenever there's a discovery like that, they like to embellish the whole thing, right? Okay, that's my theory about this. But anyways, so the linkage hypothesis, which was postulated by Morgan, which I just told you about, 2 alleles seem to be going more often than the others, doesn't tell you how this process happens. It's only when the student of Morgan figured out that there is a crossover that happens on the DNA that they kind of figured out why it was happening. So if you look at the DNA under, if you use spectroscopy for this, you will see that sometimes it will have these, it kind of binds to each other, okay? And then just switches. And these places here where the arrows are is called a chiasma. Well, all 3 of them, chiasma, okay? Just some theory for, I don't know, okay? Usually these crossovers don't happen often. As you can see, in the whole thing only happened 3 times. Make sense? Good, okay. So the crossovers, the way they happen is that when you have your duplication of chromosomes, then you produce these sister chromatids, and then these two are sister chromatids. And then crossovers happen between non-sister chromatids. So this one with this one. They still happen between sister chromatids very rarely, but they don't change the genotype pool, because you're basically having a crossover between PR plus VG plus and PR plus VG plus. So it doesn't affect the genotype. You don't see it, right? And it can happen between any of the chromatids. So this top one can cross over with the bottom one or the third one, ainsi de suite. Okay? Conventions. I know Isabella loves conventions. Okay, more conventions. So we talked about conventions between genes that are on different chromosomes. Now when they're on the same chromosome, we have to talk about their position relative to each other again, but on the chromosome, okay? So if the dominant alleles are on the same chromatid, like this, we call it cis configuration, okay? So cis means adjacent, or similar, or whatever, okay? So in this case we have a cis configuration, okay? And the way you write it is big A, big B, / small a, small b, or this plus notation, okay? For the trans configuration, the dominant alleles are on different homologs. So in this case, you'd have big A, small b on one chromosome, and the other one would be, or chromatid, and the other one would be small a, big B. So this is called trans configuration, okay? Does that make sense? Okay, so probably in the questions for the book or tutorial, you'll have something saying the two alleles are in cis or trans configuration. And it's important for you to know, because you need to know where they are relative to each other, so that you can do the crossovers properly, right? Okay, good. Yep, yep. Right. So actually there's regions in the DNA that are more prone to crossovers, because they have these repetitive sequences that kind of are easier to bind with the other side. And so there's regions that are less sensitive to crossovers, and so crossover almost never happens there. Okay? Yeah. No. Happens by itself. Yeah. Very almost random. Sometimes you have a crossover. Sometimes when a crossover happens in this region, it will prevent a crossover right next to it. So, but it happens by itself. Like DNA just does it by itself. Same thing with mutations, right? The DNA replication happens, and then mutation, just because of the wrong copy, nucleotide was added there, then you have a mutation, right? But yeah, we'll get into the DNA replication stuff, which is my forte. This is like the intro. It's been a long time I didn't do this. But since my research is more focused on DNA replication and proteomics and all that, it's going to go a bit more smoothly. But yeah, theory. Let me just drink because I'm dying. I was sick this weekend. Okay. So when genes are on the same chromosome, usually they are linked, okay, depending on the distance between them. Again, I'm repeating this like 30 times, but if they are close to each other, they will probably assort together. If they are far, no. Yeah, exactly. So if they're close together, they will go together. If they're far, they will not go together. They will act as if they were on different chromosomes, let's say. Okay, same inheritance pattern. Okay. So in the case where we have this kind of configuration for the chromatids, then because the genes are linked, we're going to produce these gametes on the right here. So big A, big B, and this one will end up being big A, big B, same thing. Small a, small b, small a, small b. And then when they are linked, something called the crossover happens. So when you have your two chromatids like this, what happens is that they open up in between the two genes. Crossover happens, and then they exchange alleles. So in that case, you end up like this. Okay, so big A, big B, and then small a, big B, big A, small b, and small a, small b. And then the chromosomes leave. Okay, they're separated by the spindle and then segregate into our gametes. Is this clear? Okay, good. So that was the simple part, I guess. Let's talk about the more complicated part. Okay, so usually crossovers, there's 1 to 2 that happen, sometimes 3, but most of the time 2 that happen. So obviously you have to add in a third gene there. And then when you add a third gene there, the distance between the 2 genes, if it's a longer distance, for example, between gene B and C, then you have more chances of crossover happening because you have more DNA available for the crossover, right? If they're closer to each other, you have less frequency of these crossovers, right? And then, as I said earlier, the crossover can happen between any of the chromatids that are non-sister chromatids. So number 1 with 3 and 4, number 2 with 3 and 4, and then 3 with 1 and 2, etc. Okay? So these are all the possible ones. And what did I want to say? Right. So then we're able to use the percent of recombinance because the longer the distance between the genes, the more recombination happens. The more recombination happens means means we can actually use the distance or the percent of recombination to figure out the distance between the 2 genes, right? Because if it's longer, you have more recombination, okay? If it's shorter, less recombination. So it's directly proportional to, they are directly proportional to each other, okay? Everything clear? Am I going too fast? No? Anybody? Yeah. Okay. So when 2 alleles are close to each other, they have more chance of going together. If they are separated by a long strand of DNA, there is a possibility of crossover happening between them, and then they will not go together. So the more distance you have, the more chance of recombination happening. And so we can use the frequency of recombination as a measure for the distance between 2 genes because they're directly proportional. Make sense? Where did the question come from? I don't know. I'm looking this way. I guess it's this way. Okay. Yeah. Sorry? Exact, yeah. Each one will end up in a different gamete at the end. Okay, so these 2 are the same and these 2 are the same. Make sense? Yeah? Chromatids are makes sense logically, but you have to imagine the DNA in like a 3D structure and like all enmeshed, and it's not that linear, right? Yeah. But yeah, of course, if they are far from each other, it's harder to do the crossover than if they're close to each other. But in this case, it doesn't affect it because it's enmeshed together and it's like a spaghetti in there, okay? Yeah. Any other question? Yeah. It's just a show where the crossover happens. That's pretty much it. So it's happening between gene B and C of chromatid 2 with gene allele B and C of chromatid 4. That's pretty much it, right? When we're talking about different, wait, let me just go backwards. So here, usually they show them in this type of color, right? The 2 sister chromatids in one color and then the other is in a different color, right? But it's basically the same thing. In this case, they decided to highlight just the crossover itself, okay? Oh, I see. I don't know, man. The design guy was having fun. I don't know. I think technically it should be different colors. Like this one here should be one color to show where the crossover is happening, and this one a different color. So this one makes sense. Yeah. So this is like starts here, and then going up here and here, right? This one here seems to be a bit not clear, but even then it's kind of clear. Yeah. Yeah. You end up with the same. No, no. You close. You end up with the same. So, if you look at the distance between A and B, right? There is less DNA. So there's less sequence for the two strands to bind and then switch. Okay. Here between B and C, there's a longer stretch of DNA. So it can happen here, 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 right? But you still end up with the same configuration between B and C, no matter where the crossover happens. So when the gametes are being produced, right, and then fertilization and all that, you'll have more progeny that have a crossover between B and C than between A and B, right? Exactly. Yeah, exactly. So you see how, so let's say we had this one, this part here, replicated 4 times, okay? So the crossover can happen here, it can happen here, it can happen here, but it's all giving you the same thing. While in A and B, you can have only maybe a crossover here. So the progeny will be representative of that. And it will have more recombinants between B and C than A and B because of the distance allowed for the recombination, okay? The further you go from each other, the closer you become and you assort independently. Okay. Yeah. Sorry. Can you repeat that, please? 2 different genes. Well, this is what's showing up here, right? No, the crossover is happening between B and C. Understand? Yeah. So you're starting with A, and then B, and then you're getting this small c instead of the big C. See what I mean? Like a double recombinant? I'm not sure I get your question. Ask me after, buddy. Yeah. B and C? Yeah. Yeah. No. Yeah, because the crossover happens with similar sequence, right? So it has to happen at the exact same sequence, not like you're getting 2 copies of one gene, and right? Yep. Two versions of one gene and the other one has 0. How can a crossover not happen at the same place? Because you need similar DNA sequences. Yeah. Send me your teacher's email. No, but I mean, there's always mistakes happening. So for sure, if the because chromosomes align pretty well, okay? Let's say they don't align pretty well. You can have a crossover between one side and the other, but this is like mega rare. Okay, it's almost impossible. And if that happens, you end up with like bad DNA, and the cell will die. The gamete will die. It cannot live. Okay. Usually they're aligned pretty well, and for a crossover to happen, the sequence needs to be the same on both, right? You can't just have like a random break and join. But I guess it happens in nature, and then the gamete dies or something like this. Okay. Yeah. Yeah. It's because they are aligned together. Yeah, exactly. You can't because for them to kind of rejoin, you need a specific like sequence to rejoin with the other one because they form bonds between each other, right? We'll see this in like chapter 7, I think, or so. Okay. Yeah. Exactly. Yeah. So let me actually like do something on this board, my favorite board there. Show you how it is before, okay? So we can kind of understand what I'm saying. So imagine you have chromatid number 1 and chromatid number 2, and then A, B, C, and then let's say here you have the recessive ones, okay? I'm flipping the diagram. So what happens is that, let's say in this region here, there is breaking. Jeez, this is so sensitive. There's breaking that happens here, and then you end up linking this one with this one, okay? And so what you end up with is this kind of configuration, okay? And then this one will rejoin with this guy here. So you end up with this possibility here. Does this make sense? Little c, yeah. Good. You guys are following, man. Okay. Does this make sense? Okay, now imagine just this 4 times. So 4 chromatids, and then they'll all separate in different gametes. So this is gamete number 1, 2, and then you'd have 2 more. Easy? Okay, good. Let's get back to this. Okay, so again, I just said that the distance between 2 genes, the longer it is, the more recombination. So they're proportional. So now we can kind of count the distance between 2 genes. And this guy called Sturtevant, the undergraduate student of Morgan, figured out this rule in one night, apparently. And so he decided that 1 genetic mapping unit is equal to 1, means 1 product of meiosis in 100 will lead to recombination. Okay? So again, I said the frequency of recombination is the distance. So 1 product of meiosis in 100 is recombinant. So then if you have 3% recombinant, that's 3 out of 100 that are recombinant, right? Okay, good. So let's imagine these 3 different genes, A, B, and C. Then if we say that there's 5% of the population of the offsprings that are recombinant between A and B, so then we can say that it's 5 mapping units. Another way to say mapping units is centimorgans. So it's just a way to call these different things, okay? And then, so if we were to do the numbers for this, we would find like the parental being like, let's say 35, and then the recombinant between A and B would be like 5%. So, I don't know, 5 out of 100, okay? Make sense? If we're looking at A and C, we see that it has 3 mapping units. So now if we try to determine the distance between like the map of the 3 genes, there's 2 options, right? We have either the gene A in the middle or C in the middle, because you can have 5 plus 3 gives you 8. So C and B are 8 units apart. Or we can have them inside the distance between A and B. And so you would have C and B at 2 mapping units. Is that clear? Yeah. Right. So let me get back to this here. So you see these numbers here? So let's assume this is A and B, okay? Right? So let's say the total here is 100. You would find this one at 2.5 and this one at 2.5 for a total of 5. Right? So that's the distance between the 2 genes. We'll see it in a second with the 3 genes, but basically 5 mapping units means 5% of the recombinants between those 2 genes in the progeny, right? Make sense? No? So, yeah, progeny is recombinant for those 2 genes. Make sense? Kind of? Yeah. Questions? No? Okay. We're going to get into the 3 genes and then it's going to go crazy. So if you have a question about this now, it's good to ask it. No? Okay. Yep. Right. So both. So they're both proportional. So when you get a percentage, so when we talk about the recombinant frequency, it's the frequency of the progeny that will be recombinant for those two genes. Make sense up to now? Okay. Because they are both proportional to each other, meaning that the longer the distance between the 2 genes, the more recombination happening, the bigger the recombinant frequency, and the more recombinant offspring, so then we can use it interchangeably, okay? But it's just arbitrary units for the mapping unit. Like it's not something, right? It's not precise. We actually use base pairs to count. It's just what they did all time ago, okay? So just remember, 1 mapping unit, 1% of recombination frequency. Okay, yep. Yep. Same. Yeah. Yeah, because the progeny, so the recombination frequency takes into account 2 genes, no matter where they are. It doesn't take the length of the chromosome. If you're looking at 2 to 3 genes, you would have to do it 2 to 3 genes at a time, right? You can't just go from one to the other, right? And obviously, you're pretty smart because you thought about if the chromosome is this big, that means are we going like 200% mapping unit? No, you have to think about it as in the relationship between 2 genes and irrelevant of the length of the chromosome. But yeah, that's a good point. What we use actually now is just the base pairs, the number of base pairs. Okay? Okay. Let's get back to... Okay, time to complicate it with 3 genes. Are you guys ready? Okay. I was actually thinking about how to explain this because it's not complicated, but might look complicated, okay? So I forgot who did this cross, but let's just say somebody did this 3-point test cross, okay? So he took one progeny with homozygous for V gene and then double recessive for CV and CT and crossed it with the opposite. So that means the gametes produced are V plus CV CT. Makes sense from here. And then V CV plus CT plus. And then you form your trihybrid by doing fertilization. Up to now, same thing that we've been doing, okay? When you get a problem like this, which I can guarantee you will get, is you'll get something like this and then a table like this, okay? And then you have to figure out what gene is in the middle and the distance between the genes, okay? So this is the process through which you do that, okay? So first thing you have to realize about this is that the tester male, we forget about it, okay? Because the tester male will be useful in doing the Punnett square, but we are thinking about the gametes right now. So we just take the trihybrid female because the trihybrid female is the one that will produce the gametes that are interesting for the phenotypes later on. Okay, make sense? And so you end up with 8 different gametes, and then you have 2 of them that are a high frequency. These are the parental. Right? So this is the whole population of the gametes. You end up with the 2 that are the parental. You end up with 2 others that are at an intermediate frequency. You get another group of 2 that are at a different intermediate frequency, and then you get 2 of them that are at a very low frequency, okay? These 4 here are the single recombinant, so there was one crossover that happened, and it's between 2 different genes, so these 2 happened within the same gene, the same 2 genes, and then these 2, it's a group of 2 other genes, okay? These last ones are the recombinants are double recombinants, so that means there were 2 crossovers happening between V and CV and V and CT, okay? Make sense up to now? Yeah. How do I know that? Yeah, so the parental, because they're linked, the parental will always come to the top, okay? Why? Because it's more plausible to have them go together than for a recombination to happen. Recombination doesn't happen often. So the parental would be the highest. Now the double recombinant, you need 2 recombination events to happen, which is even more rare. It's the probability that one of them happens times the probability of the other one of happening. Does that make sense? So these ones are the lowest of them. And then these 2 and these 2, how do I know? Well, you just need to learn it like this, but I will explain in a second. But basically, the numbers that look alike are probably for the same 2 genes, and then the others that look alike are for 2 other genes. But you'll see it in a second. Okay? Yeah. You don't. What order? The genes or the table? Something similar to this. Something similar to this, but you'll have, again, 2 of high frequency, 2 of very low frequency, and then 4 of them of intermediate frequency. 2 similar and 2 similar. Okay? Yeah? Exactly. So you can imagine that these 2 have a bigger distance between them than these 2. Make sense? Okay. We can't say that for this one because double recombinant. Okay? But we'll come to this. So when you get a problem like this, you'll probably be asked to figure out the map, okay? Because we're into making maps today, and not the BS map that we saw earlier, okay? A real map. And so the first thing you need to figure out is so you'll end up doing something like this, okay? And I'll get back to this slide. But the first thing you need to figure out is the gene in the middle. And to find the gene in the middle, you need to look at the parental and the double recombinant. And you need to compare them. And the gene that's different between the double recombinant and the parental is the one in the middle. Let's see, for example. So the parental, we have VCV plus CT plus, VCV plus, but this one is CT, right? Does the other one have VCV plus? No. So the gene in the middle is CT. Easy rule to remember, right? Yep, I'm repeating. Okay. Let me actually draw it. You know what? Let's do this. So it's going to be easier. Where's this pen? Okay. So the parental is VCV plus and then CT. We don't know the order yet, okay? I'm just putting it randomly. We'll see like how you get the order, okay? The other parental is V plus CV CT. V plus CV here like this, and then this one like this, okay? Does this make sense? Okay. Now if we look at the recombinants, how can we get V, CV, CT? V, CV plus, CT. How do you get that? You need a recombination right here, right? Okay, but we have to consider the other one too, right? For the other one to happen, to get V plus CV, CT plus, what am I saying here? Wait, give me a second. No, I'm messing up my whole thing. 1 second. Actually, I think I have slides for this. There you go. I have slides. Let's get back to the slides, and then I'll redraw. Okay. Okay. So this is it. Okay. So you can have 3 possible gene orders. Let's start by this. I think it's better. So either the CT gene in the middle, the V gene in the middle, or the CV gene in the middle. Okay. If we have a double recombinant, so these guys here, right? We can only produce these 2 chromatids because you'll have V, CT, CV plus. Does that make sense? Okay. And then I don't know what I was doing here, but forget that. Okay. And then we can also have V plus, CT plus, CV, right? V plus, CT plus, CV. Okay. If we had V in the middle, we'd have these combinations, and if we had CV in the middle, we'd have these combinations because of the double crossover. Does that make sense up to now? Okay. So if we compare the double recombinant to our options that we got, right? So it's V, CV plus CT, V, CV plus CT, right? So it's this one here, okay? Here the genes are not in the right order. And this is just for you to figure out because I can't give them to you in the right order and then ask you to put them in the right order, okay? So you have to assume that they're not in the right order, okay? So the only way for you to get this genotype here, wait, let me get my thing. The only way for you to get this genotype here is by having CT in the middle. If we're talking about double recombination. Am I clear? Kinda. Did I lose you? Starting to lose myself here. Yeah. Sorry. How do I know if it's a double recombination? Sorry. Okay. Repeat that, please. If I ignore the numbers? Yeah. You can. You need the numbers. Yeah. Because the numbers will tell you the frequency of the progeny that have that genotype. But obviously, if we switch it and that frequency would be higher, then it would be a single crossover and then the gene order would change, right? But here, because we know that it's the smallest group, then we know that these are the double recombinant in this kind of example. Okay, I will repeat that one more time, just in case. The first step that you have to do in a 3-point test cross when the genes are linked is to find the gene in the middle. To find the gene in the middle, you first have to remember that the order that you have here is not the right order. Could be the right order, but not always, okay? So you have to ignore the order here. What you're trying to replicate is the genotype right here, okay? So you will take the parental, and then you will try to figure out how you can get from here with a 2-time recombination. So you have 3 possibilities, which are the 3 genes in the middle. CT, CV, and then CV, okay? So when you do all these, you end up finding that if you have a double recombination, you will end up with these sister chromatids, and so this is the right configuration, okay, with CT in the middle, because it's the only one that can produce V, CT, CV+. V, CT, CV+. Okay? Yeah? No. No. That's the middle one that's more important, because you can just flip it and it's the same thing. Okay? Make sense? Okay. Good. So the top one is the right one. Okay. So the mistake that I just did here when I was drawing, I took it as this being the right sequence. Okay. So exactly the mistake that you might do on the. So don't do what I did here. Okay. Yep.
So the parental one.
Yep.
That's the right one.
No, even this is not the right order. Yeah. Just we're looking at the genotype. The order, we don't know if which one is in the middle. Okay. So what I do when I want to find the gene in the middle, I compare the parental with the double recombinant, and I find the gene that changes, because that's the gene in the middle of a double recombination event. Okay. So here we have VCV plus, and then just the CT that changes. So CT should be in the middle. If it was, yeah.
So it doesn't matter like which parental you use.
Yeah, as long as the same gene will be the one that changes in both. Okay, next step, the distance. So for the distance, you have to think again, just like we learned before, 2 genes at a time. Don't start thinking about all genes together, okay? So if we take the gene VT, V, and CT, okay? And we look at the parental here, and we compare with the other progeny, we look for the one that there's a recombination between V and CT. So if we look here, we have V, CT plus, and then suddenly we have V, CT. So this is a recombinant for, there's a crossover that happened between V and CT. Do you see that? Okay. And then same thing here, because we start with V, CT, but now we have V plus CT, and then we end up with V plus CT plus. So there's a recombination that happened between V and CT again. And obviously the double recombinant has a crossover between those 2 genes. Does that make sense? So we find those, we add all the numbers of the progeny that have a recombination between V and CT, divided by the total gametes, and then multiply it by 100, okay? And that will give you a percentage, which is the recombination frequency. This recombination frequency can be converted into mapping units. So we can just say 13.2 map units. Okay? Yeah? So the crossover can happen either between the gene V, CT, or between CV, CT. Or between V and CV, I mean. Okay? So then when I'm asking you where, to figure out which of the progeny have a recombination between the gene V and CT, you look at the parental between V and CT, and then you look for the ones that have a change. No, we'll look at the other one too. The other combination. We'll look at each combination of genes. Okay? You get it? No? Okay. It's okay, we'll get it, buddy. Okay. We're trying to measure the distance between V and CT. Right? So to find the distance between V and CT, we have to look at the parental unit and then compare it to the recombinant and find the ones that have a recombination between the gene V and CT. Does that make sense up to now? On the DNA strand. So the crossover happening there. Yeah. Yeah. Makes sense? Yeah. Right. So should I erase this? Yeah. You know what? Let me try to do something different here. Okay. I'll just give you an example, then I'll put it back. So if we're looking just at V and CT, okay? So you go like this. Okay? So these are the parental. And then you look at all the other offsprings, not offspring, but all the other recombinant possibilities, and you compare the V and CT genes alone. Okay? So the parental has V, CT plus, or V plus, CT. Right? So you look for the ones that are different from this here. Those are the recombinants. That's where the crossover happened. Okay? And then you count those, all of those. You divide it by the total number, multiply it by 100, and it gives you a recombination frequency, which is this 13.2%. That's 89 plus 94 plus 3 plus 5. Opposite. 191 divided by 100, 1,448 times 100 to find the percentage. Make sense? Okay, cool. Now, do you get how you find the ones that had the recombination event between the genes V and CT? Is that clear? Okay, cool. So let me just Control Z all this. And then go to the next thing. Okay, now we're looking at the other two genes, CV and CT. Okay, so to look at the CV and CT, we remove the V because we don't care about it anymore. Like this. And then we look at the table to figure out the ones that had the recombination event between CV and CT. So if we look at the parental here, here's my laser. The parental here, this is it. So we have CV plus, CT plus. Anything different from this or this is a recombinant, right? So CV, CT plus is a recombinant, CV plus CT is a recombinant, and then the double recombinant. Okay? So now the distance between CV and CT can be counted as follows. You count all the recombinants, 45 plus 40 plus 3 plus 5 gives you 93, divided by the total times 100. So it gives you 6.4% recombination frequency, which can be converted to 6.4 mapping units. Okay? In other words, this map. So we found that CT is in the middle. Now, when you're looking at this, this is not the alleles, okay? Don't expect any plus here or whatever. This now I'm talking just about the gene, not the alleles, okay? So this is the map of genes, and the distance between the gene V and the gene CT is 13.2 mapping units, which is the one we found here. And then the distance between CV and CT is 6.4 mapping units. So then from here, this is the part where I'm going to lose everyone, okay? Get ready. So then if you want to count the distance between V and CV, you add both, right? So that gives you 19.6, technically. The problem, the problem is that if you do what I just told you for the genes V and CV, you will find 18.5%, okay? And this is something I don't want to teach, but the book teaches it this way, so I have to cover it so you don't mess yourself up, okay? When you find your middle gene, you find the distance between the middle and both genes, right? Forget about the frequencies, just count it as I told you like this, 13.2 plus 6.4, okay? Don't count the numbers, all the recombinant numbers, and then divide it, okay? And it will be wrong. Why? Because they expect you, because this one here is not a recombinant for V and CV, right? But technically it is. It's a double recombinant, so you should add it twice. But I don't want to get you mixed up. When you want to find the total distance between the two extreme genes, just find the distance between the middle and the one on the right, the middle and the one on the left, and add them, okay? Forget about the other stuff, okay? It's something that was like wrong in the way they made this law, let's say, and then they try to explain it in a cheap way, so it doesn't make sense, okay? So just forget about that. Count the distance between two genes, the others, add them together, and that's it, okay? Good. Not bad. Not bad. Okay. Questions about that? No? Okay. Yep. Wait, the numbers is what? Sorry. Yeah. Yeah. Yeah, yeah. Exactly. Or I could give you the percentage and then just invent numbers, right? You can say, for example, like instead of it being 45, 40, you could say, I don't know, 50 and then 35, whatever. Yeah. Yeah. I'm probably not going to give you this. You're going to have to figure it out from figuring out this one and this one, and then adding them together, okay? Does that make sense? Okay, cool. Yeah, okay, it's not bad. We got the math, almost done with the math. There's the chi-square that we have to do next lecture, and then from there it's pure like learning by heart, okay? DNA replication, gene interaction, all that, okay? Good. So, few things, not bad, I'm getting better with time. Okay, so we learned a few different ratios, okay? This you need to not learn by heart, but now you should be able to understand this, right? So if you have a monohybrid test cross, monohybrid, so heterozygous with a test cross, you'll end up with a 1-to-1. If you self-it, you end up with a 3-to-1 ratio. If it's a dihybrid test cross, it's 1-to-1 to 1-to-1. If it's selfed, it's the 9-to-3 to 3-to-1. So these are all on linked inheritance. And then if you're doing linked inheritance, then you'll end up with, if it's a dihybrid, that is test crossed, you'll end up with 2 parental and 2 recombinant. That's like the first few slides we saw today. That's if they're linked. If it's a trihybrid that's test crossed, just add more ones. So 1-to-1 6 times, I think. Wait, can't read this. 6? 8. 8, sorry. 8. Yeah. And then if it's a trihybrid that's test crossed, you will see what we just saw right now. 2 parental, 2 double recombinant, 2 and 4 single recombinant. Make sense? This is what they mean by PPSCO, single crossover, and then double crossover. Okay? Good. Any questions about lecture 5? No? Okay. Back to this guy here. So, so for the red-green color blindness, these genes are found on the X chromosome, as I just showed you. So you have 2 genes called, it's the opsin, it's called opsin, long wave, and then you have also the medium wave, and long wave and medium wave are for the red and green, and they're linked. They're on the same chromosome right next to each other, right? And so let's look at this guy first. Just 1 second. Oops, sorry. 1 second, guys. OPN1 MW. Jeez. Okay, so zoom out. Maybe 10x. So you see, where is the other guy? Wait, let me just find it. Oh, right there. So see how close they are together, right? So so these opsins are found in your cone cells for your eyes. If you want to know what cone cells are good for, you can either take neuroscience at the 3rd year or mechanisms of development, the 366. You learn about this. But basically, it's the eyes has 2 types of cells. It's the ones that get the color. Okay, so whenever you have a mutation in one of these, you will probably inherit. If you have a mutation in both genes, you'll probably inherit it together. And because you inherit it together, you have the 2 mutations that you end up not being able to differentiate red and green. Okay? If they were on different chromosomes, you'd be lucky sometimes to get one good and one bad. Okay? But this one, because they're so close together, then you end up inheriting both mutations almost all the time together. That's why it's red-green color blindness. Now, why is it more prevalent in the male? It's because it's on chromosome X, as you see right here. And if you're a male, you only get 1 copy of chromosome X, and so you'll end up having the issue. Okay? If you're a female, you could get 1 copy that's healthy and the other non-healthy. You could still be good for this. Okay? And this can be developed with age. So that's why every time you go see your optometrist, they give you this test to figure out if you have it or not. Apparently, there's apps now that you can use, but yeah, that's pretty cool. Sounds good. I think I'm gonna leave lecture 4 thing for next time. I'll let you go. Thank you for listening. See you later.